Prego, cominciamo questa.
Let us now start this session with the speech of Dr. Vornoli that is linked to the previous speech, because we've seen that the holosome or the hologenome are influenced by external factors, environmental factors. You heard the floor. First of all, good morning everybody. I would like to thank the President and Professor Giuliani for inviting me here and speak about our research. I am a researcher of Ramazzini Institute of Bologna. I replace my director, Fiorella Berbaggio, that has other commitments today, but as Professor Giuliani, I will speak about something that is quite cross-cutting. I will speak about the contribution of in vivo experimental research to understand carcinogenic risks. First of all, I would like to speak about our institute, that its name comes from the name of the father of occupational medicine, Bernardino Ramazzini. Our research center is quite special because we have a privilege, because we can work in a historic place that reconciles the history with present. It is a castle with frescoes, as you can see, and there we have our laboratories. So it is a quite unusual environment, and you have a science and a history that go hand in hand there. I will present to you my institute. It is an independent cooperative, a non-profit cooperative that is financed by public, national and international funding, and also contributions from NGOs and private citizens, and the latter are over 300,000. There are 300,000 associates that each year cover 30% of our budget. I work principally in those strategies aiming at preventing environmental diseases, in particular cancer. We all know that it does not depend totally on the exposure to the environment, but the environment is very important for cancer. Our aims are cancer prevention through a strategy based on the promotion of scientific research, training of specialized personnel that are young researchers, spreading information on environmental and occupational carcinogenic risks, and also the implementation of programs, because also we have a clinic for the early detection and prevention of tumors. Our research is supported by a scientific committee that has very important members, starting from the president of the National Institute of Environmental Health Science of North Carolina, Linda Brumbaum, Pietro Comba, that is the director of the Unit of Environmental Epidemiology of the National Institute of Health in Italy. Every year, these people that are highly important gather with other people and also with us, the researchers, and they take part in the Ramazzini Days, the Genetica that are held in Carpi. So now, moving back to our main work, over 40 years we've studied more than 200 compounds, especially in SD rats, and the lifespan of the experiments is of at least 130 weeks, and in order to do that we have a laboratory that was established from the 1960s, 1970s, and we've studied more than 200,000 animals. We've studied over 200 compounds, and so because of that we are the second research center in the world for the number of substances studied. There's a brief list of the substances that we've studied and of those that have resulted to be carcinogenic, borderline, or not related to cancer at all. Our peculiarity is the human equivalent model, that is the Sprague-Dawley rat. What are the characteristics that make it valuable for the identification of the risks for human beings? First of all, many genetic, pharmacological and toxicological and carcinogenic responses are similar to the human beings, and also the reproducibility of experiments conducted independently in different laboratories in the US, for example, and here in Italy. And then the compounds that proved to be carcinogenic in humans are equally carcinogenic in rats when the research is carried out properly. These are just small examples that show that our colony culture is in the average in terms of survival compared to other great facilities in the world. This small table shows that the rat and the human being share more or less malignant cancers. As for the higher percentage of their occurrence, more than 80% in the age group that is over 65 years of age. So this shows that all the studies that stop after 104 weeks, that correspond to roughly 65 years of age of human beings, do not take into account most cancers that occur among human beings. But our studies go on for longer. This is our study concerning silene. Up until 112 weeks, there had been no significant differences between the black column and the red column, but then the difference has increased up until 95 years of age for humans. We've seen the same thing with male concept during one of our studies, and this is related to thyroid cancers more specifically. It's not really important to study those tumors after a certain age, but also the beginning is very important. This is one of the most common compounds, that is vinyl chloride. When a newborn are exposed, so from the first day of life, and when adults are exposed in the same way, 20 weeks. In this case, there are the mothers. So the starting point of their exposure of vinyl chloride shows these two different concentrations. And we can see that the neonatal exposure prevented the occurrence of such cancers, and that was not visible in the mothers, because 20 years after that it was discovered that metabolization of vinyl chloride was carried out by a cytochrome that the newborn did not have yet. These are just some of our studies. That is the predictability over the years of the based on the YAC definition. One is is a human carcinogens. As you can see, there are only one. There's a difference in the prediction years, and that shows that our studies are very predictive, starting from vinyl chloride that has only 5 years of difference in the YAC, and then in other cases such as benzene, for example, there are even 33 years of difference. Then there's asbestos in all its forms that we studied in 1989. After 23 years, it was defined as a carcinogenic agent by the YAC. And there's a formaldehyde that fluoridinite, that is something that we found out in 2017. I would like to now speak about one of our recent studies. That is a study on the radio frequency radiations. It is a field that is between very low frequencies and infrared. These are non-ionizing radiations. We focused on something in particular. This started with NTP. At the same time, NTP studied cell phones, that is the total body effects of mobile phones, where we studied the effect of the antennas of mobile phones. This is just a table that shows what we did. They focused on the near field, that is mobile phones, while we focused on the far field, that is the effect of antennas for the radio frequency of mobiles. So these are two studies that started at the same time, very far away geographically, one from each other, one for the other, and just to show you the kind of exposure of the rats in our case, and rats and mice in the case of NTP, there's the SAR value that is different, that is the value that defines body absorptions of radio frequencies by animals, and our values, a higher value was amounted to a minimum of 10 times lower than NTP, and our lower value is 50 Hertz. That was 1,000 times lower than the exposure of the NTP rats. This is our exposure system that we created to this end. Rats were placed in 4 different rooms. In each room was insulated, which means that this device, I cannot remember the material of it, but this device allowed to prevent a non-uniformity effect in terms of field. So we did not use normal walls, we used insulated walls, and rats were placed in these wooden structures. So there were no other materials that could interfere with the radiations, and the same applies to the cages. And at the center we placed this antenna, which basically, as you can see, reproduced the exposure like the exposure of citizens that are exposed to an antenna that is close to their house. So that was the idea. And each one of these antennas had a different intensity of frequency. The speaker corrects himself of intensity. Very recently, NTP and us have published, after some years, the results of these 2 studies that were carried out separately, but at the same time, as I told you, I also had the honor to participate in the National Toxicology Program through a fellowship. Our work was recently published by Environmental Research, So we showed that. Okay, I'm trying to be brief because I'm running out of time, but we showed that we noticed something different from the NTP. NTP noticed an increase in hyperplasia in the glial cells in male rats, where we early saw a slight but not significant increase that is dose-related in malignant glioma in exposed female rats. While our most interesting discovery is the significant dose-related increase in malignant schwannoma that was noticed by the NTP2, and also we noticed a not significant slight increase in hyperplasia in both males and females. Talking about schwannoma in males again. This is an example of schwannoma found by the NTP, and that is the one of the Ramazzini Institute. And you can see the typical aspect of cells that characterize schwannoma. So we can say that our outcome is quite in line with the NTP outcomes. This kind of tumors of the brain and the heart have the same cytological origin of those observed in some epidemiological studies on cell phone users that are male users. This information might be enough for the ARC to review the category of radiofrequency. We aim to finish the evaluation of all organs, the review of all organs, and also we want to conclude the mechanistic studies on heart and brain tumors. I focused on the target of resequencing of RNA of tumors, the tumors of the heart and brain that I showed you earlier, based on previous studies carried out with NTP that showed that our fixation method of organs, that is alcohol, and because of that we're different from the other research centers of this kind which use formalin instead. So whatever the duration of the fixation of the organ, they did not influence on the quantity and the quality in this case. I'm showing the nanodrop data for the DNA and RNA. I went to the United States and focused on this project and to prepare libraries that have, and now we are in the sequencing phase, so we're waiting for the outcome. Here you have the characteristics, but I think I have no time to speak about our protocol. The fundamental thing is that this kind of protocol of Illumina can be perfectly adapted to the samples that are fixated in alcohol, and the DNA input to prepare these amplicons can reach 10 nanograms, so it can be adapted to very small amount of DNA that we can extract from tumors at the times that are very, very small. Here you can see the reading depth that allows us to identify pre-neoplasia mutations. That is the list of genes that we studied. These are the genes that were found as to be mutated in the rats, and we want to compare them with the human ones. Some have all exons that were mutated, some of them had only some mutated exons. Here is the description of the protocol, but anyway, we analyzed the libraries of the DNA obtained from the qualitative point of view that is ideal. These are examples of our samples. Here is the list of samples. That is the heart of one of our animals. We were expecting a product after the amplification of amplicones with a kit around 300, and that's what we had. We can see it in the tape station and the PCR. So as we're waiting for the final results, final outcomes, and thank you for your attention.
Grazie, dottor Vornoli.
I would like to thank Dr. Vornoli. Now there's a lecture that is very interesting, that shows how an ubiquitous pollutant, such as ionizing radiations, can have an unexpected impact on genetics too, of our body, or anyway, of the experience